Today we are going to discuss a very important topic, that is food poisoning. This is a very important entity, especially encountered in the rainy and the summer seasons due to ingestion of contaminated food and water. Now coming to food poisoning. Food poisoning is referred to an illness acquired through the consumption of food or drink which is contaminated with microorganisms or their toxins. Bacterial food poisoning refers to acute gastroenteritis due to presence of bacteria or their toxins in food. This generally results from consumption of food at a common source in the form of an outbreak. Sometimes non-bacterial causes are also responsible for food poisoning, like chemical poisons and mushrooms which may be sometimes poisonous in nature. Food poisoning may be classified into three categories. That is the infective type, toxic type, and intermediate type. In infective type, the infective doses of microorganisms are ingested with food and multiplication of bacteria occurs in vivo. In toxic type, preformed bacterial toxins are ingested in food, and typically it is seen in staphylococcal food poisoning. The incubation period being from 1 to 6 hours. In intermediate type, the bacteria is ingested with food. And on reaching the gut, it releases the toxin. Example: Clostridium perfringens food poisoning, and the incubation period is 6 to 12 hours. Food poisoning may be due to variety of factors like improper storage, inadequate cooking, eating raw vegetables and food. Also, if the food is cooked by carriers or infected persons, they also contribute to food poisoning. In infective type, the causative agent generally includes Salmonella typhimurium, Vibrio parahemolyticus, and Campylobacter jejuni. In toxic type, the responsible organism is Staphylococcus aureus, Bacillus cereus, and Clostridium botulinum. In intermediate type, the organisms responsible are Clostridium perfringens, Bacillus cereus, especially the diarrheal type. Now coming to the pathogenesis, the infection occurs by consumption of food. Like for example, vegetables, salads, poultry, eggs, fish, meat, seafood, fried rice, raw milk, and milk products, canned food and cereals. Mechanisms include the invasion of gastrointestinal tract with bacteria, leading to diarrhea, or there can be ingestion of preformed toxin in food, preformed toxin in food, as in case of Staphylococcus aureus and Bacillus cereus, which forms the toxin in food before it is consumed. Before it is consumed. This leads to vomiting and diarrhea. Also there can be ingestion of bacteria in food, but toxin is produced in gut after ingestion. Example being Clostridium perfringens, which cause diarrhea. Generally, Salmonella food poisoning occurs on ingestion of milk, eggs, poultry and meat. Consumption of seafood causes infection with Vibrio, raw milk, poultry and contaminated water, causes infection with Campylobacter jejuni, Canned food causes infection with Clostridium botulinum, and fried rice from Chinese restaurants, vegetable, meat and cereals cause infection with Bacillus cereus. Clinically, the incubation period varies from 1 to 24 hours depending on the type of food poisoning. Depending upon the type of food poisoning which is characterized by nausea and vomiting, diarrhea, abdominal cramps and fever. Vomiting is rarely seen in is rarely seen in Clostridium and Bacillus infection. Vomiting is rarely seen in Clostridium and Bacillus infection, but abdominal cramps may be present, and diarrhea may be a characteristic of all food poisoning. Lab diagnosis is done by taking of detailed history which includes the type of food consumed and the sequence of clinical symptoms with their duration, and also how many persons are affected from the same source. Collection of specimens like stool, suspected food specimen, may be done. If death occurs, liver, spleen and blood may be collected. Culture is done to demonstrate the causative agent in stool and food. It is done on blood agar, McConkey agar, and certain selective medias like mannitol egg yolk, polymyxin B, phenol red and agar may be used for Bacillus cereus. The Clostridium group, that is the perfringens and botulinum, are cultured under anaerobic conditions. Next, identification of causative agent. Is done on colony morphology, on culture media, Grams staining, and biochemical tests. Enterotoxin in feces is detected by ELISA method or latex agglutination test, also polymerase chain reaction, may be used for detection of genes coding for enterotoxins. Next, identification of carriers. Identification of carriers is very important, and this is done by repeated culture of stool, and other specimens. The only prophylaxis to prevent food poisoning is to maintain a strict level of cleanliness, at all levels of preparation, storage and serving. Preparation, storage and serving.